Well, hello, my name's Ian McInnes, and it's a real honour for me to be presenting this lecture to you. Your august society studies matters which are well beyond me, namely imaging and MRI in particular, and therefore I can promise you that the next 20 to 25 minutes or so will be an MRI-free zone. These are my relevant conflicts of interest. I've received funding from a number of academic funding agencies, and I've worked with a number of pharmaceutical companies, particularly those concerned with the advent of immune-modifying therapies. I think it's also important to know where your virtual speaker comes from. I come from Glasgow, a wonderful city in the west of Scotland, a heritage I share with some very senior members of your own society. And when the travel ban eventually lifts, I would encourage you to come and visit with us. You'll have a wonderful time. It's a city of fine culture, architecture, and especially it's the home of the Glaswegian, amongst the friendliest people in the world. I promise you a very fine time indeed. Well, let me turn first of all to the source of the problem. The coronavirus superfamily contains a number of different genera. We are concerned particularly with beta coronaviruses. The SARS-CoV-2 genome is related to a bat coronavirus that may in turn have come from pangolins, believe it or not. And it's one of three major coronaviruses that have been found from zoonotic reservoirs in the last 20 years, SARS-CoV-1, SARS-CoV-2, and MERS. And these are viruses that, for now very obvious reasons, are of critical importance to us as we face one of the largest public health emergencies in more than a century. Let me turn first of all to the structure of the SARS-CoV-2 virus. On the left-hand side, a cartoon depiction of the genome and the genes that are contained therein. I want you to focus particularly on the green bar. This is the sequence that codes for a so-called spike glycoprotein. Now, if you turn your attention to the right-hand side of the slide, you'll see a schematic of the virus itself. You'll see these spike proteins arranged around the edge, giving the so-called corona-like effect, given the name and turn to the virus. And these are of particular interest to us because not only do they allow the virus to enter cells through ACE2, but also they are primary immune targets for potentially peptides that T cells will recognize and structures that antibodies that neutralize viral entry will be formed. I'm very grateful to David Bella, who works in the MRC Centre for Virus Research in my own university in Glasgow, for lending me this beautiful EM picture. And you can see in the centre one of the viruses looking particularly corona-like. So this is, if you like, the enemy. Now, we've known for many years that the immune response that occurs upon viral infection is really rather heterogeneous. And this in turn depends on numerous host factors, age, comorbidities, for example, obesity, hypertension, and also fundamental immune genetics. None of us have an immune system that is identical. It has evolved over time, and those are subtleties that emerge in our ability to respond to different infections, different insults, different stimuli. There's also increasingly some evidence that the changes within the virus structure itself may alter the way in which it engages with the immune system. As such, it shouldn't be a surprise to us that there is a balance that has to be formed between an immune response necessary to clear virus and an immune response which may be overexuberant, causing, if you like, secondary damage, causing tissue damage, and then in turn tissue dysfunction. And if that is in an organ such as the lung or the heart, which are incredibly sensitive to functional disturbance, that can lead very quickly to very severe clinical consequences. And as noted, this essential balance between clearing immunity, protective, safe immunity, and immune pathology is something that is driven by a whole variety of insults in terms of the virus and host factors, as I have described. I want to think just for a moment with you about the fundamentals of cytokine biology. Now, cytokines are, if you like, the hormones of the immune response. They allow cells to talk to each other, different leukocyte subsets, but also leukocytes to talk to host stromal cells, myocytes, enterocytes in the gut, and so on and so forth. And the immune system is fundamentally organized into subgroups of cytokines. So cytokines such as interferon gamma and IL-12 are involved in regulating an intracellular virus or intracellular bacterial response, a type 1 response. Cytokines like IL-4, IL-5, IL-33 lead to a type 2 response, evolved to kill helminth worms. Cytokines such as IL-17 and IL-23 drive what is called a type 3 response, particularly involved in fungal killing. And a rather loose array of cytokines such as TGF-beta are sometimes known as the type 4 response, involved in a whole range of microbial responses. Now, the things you need to know about here are, first of all, cytokines work in teams, not as individuals. And secondly, they tend to counter-regulate each other. And so different cytokines, when switched on, will almost always counter-regulate another. And that is how we get immune homeostasis in the normal event. Consider now the situation in COVID, and this is an image with which I suspect many of you are now familiar. It is a cartoon depiction of the severity of illness on the y-axis over time on the x-axis. And in this particular image, the idea is that the early phase will be a viral response phase where a majority of people happily will develop a protective immune response that will clear the virus and lead to relatively rapid resolution and mild symptom complexes. There is a second phase predominantly recognized in the lung, although I think we're beginning to see that this is probably much more systemic than that, when the host immune response balance begins to really matter. And in some small number of, sadly, very unwell patients, there is a hyper-inflammatory phase, so-called stage 3, where ARDS and multi-organ failure may ensue. And that's the phase that I want to particularly focus on in discussions today. This phase has been termed a cytokine storm, although I'm going to suggest to you that it may just be chaotic immune cellular dysregulation. It's the whole idea of chaos, of course, was originally described for the butterfly causing a whole range of problems on the other side of the world. And the factors that might regulate the capacity for the butterfly, that being a normally relatively mild coronavirus infection, becoming a storm, if you like, lies in host immune genetics. We know that there's an association with blood group, for example. There are genetics in endothelial cell function, coagulation function. We know that, for example, smoking and diet are likely to be very important in the way an immune response is regulated. And we're increasingly recognizing that factors such as hypertension, obesity, diabetes, metabolic syndrome, and just being old and therefore having inflammaging and immune senescence may all regulate the way in which an immune response can go from protective to potentially deleterious. Now, this has led to the concept of a cytokine release syndrome or a cytokine storm. This has not been invented for the COVID response, but is actually a well-recognized clinical syndrome, which was particularly evident in, for example, children with hemophagocytic syndromes, but also after some microbial insults. It was recognized with very tight temporal relationships with the administration of CAR T cells in oncology, where very high levels, if you look at panel B, very high levels of cytokine responses were measured in a short period of time. And occasionally there are more chronic phases of this. But I want to turn particularly to this hyper release of cytokine. Now, if you look at this figure, this is the CAR T cytokine release syndrome, if you like, the archetypal cytokine storm. And you see in the left-hand side very high levels of interleukin-6 and interferon gamma. These are pro-inflammatory cytokines. But look at this, almost several thousand picograms per mil of cytokine being produced. On the right-hand side, a very similar massive upregulation of cytokine, in this case, in patients with HLH. What are we actually seeing in the COVID response? Well, here are data from one of the earliest studies published out of China, and these are elevated levels of cytokines. I've chosen just a few from this paper that measured more than 20 different molecules, and you'll see high levels of interleukin-6, interleukin-17, interferon gamma, and even TNF, particularly in those patients who were in the intensive care unit. However, look at the y-axis. The actual concentrations of cytokine being measured are in the hundreds or even less, and certainly are not in the thousands. those being the levels that were originally described in a so-called cytokine storm. Let me turn now to a more recent study, in fact published just 2 weeks ago in the journal Nature, this arising from a very fascinating approach taken in Yale University, in which a number of patients, 33 with severe disease, 80 with moderate disease, as compared with 108 healthcare workers, donated blood and plasma for the measurement of cells and cytokines. You see in this slide in the bottom right-hand corner a heat map that depicts the level of cells, and you'll see that there is a group of patients who have high levels of monocytes, neutrophils, that being the dark color as opposed to the light yellow, and lower levels, for example, of CD4 and CD8 T cells. And that group turned out to be the group that suffered most severe disease. And that's an indication, if you like, that there may be some innate immune component to that process. I apologize for the ferocious complexity of this slide, but just stay with me for a moment. What the authors then went on to do was to divide many different cytokines that they measured in plasma into functionally related teams, a lilac team, an orange team, a green team, and a purple team, if you like. They then asked the question how these groups, if you like, cytokine signatures behaved across different patients with different age, different sex, and different clinical outcome. And they asked the computer to cluster these different characteristics into groups. And there was one cluster that came together in which very high levels of 3 of the key cytokine signatures were most obviously expressed. And if you look down in panel D, you'll see that that cluster 3 was the group of patients who were most likely to develop coagulopathy and most likely to die. Now, these are really important data because they give us a clue, first of all, that patients quite early in their disease course may already be predicated for a poor outcome, and that would give us a chance to recognize them early. And secondly, it suggests that these cytokines may even have a pathogenetically important role, allowing us thereafter to potentially intervene. I want to pause for breath now and just tell you a little more about cytokine biology. And these are lessons that we've learned from the world of rheumatology in the last 20 years. Cytokines, more than 100 of them in human biology, either they work as a linear model, cytokine A drives cytokine B, or maybe they work in parallel or even in more complex networks. Now, the original model was one that had TNF as the keystone cytokine. It drove the production of a whole lot of other downstream cytokines, for example, interleukin-1, interleukin-6. And that meant, and this is the pivotal and seminal study of Mark Feldman and Tiny Maney, that if you block TNF, everything else would get better. Now, that is a good idea if you're developing immune therapeutics, but not such a good idea if you think about it, if you are trying to develop a host defense response. The microbes would, I think, pick this possibility up very quickly indeed and would render us highly susceptible to a virus developing an anti-TNF protein. And that, of course, would then render the immune response redundant. So this is, at an evolutionary level, very unsatisfactory, although I have to say at a therapeutic level, highly successful. Because the immune system is incredibly complex. This is actually a very old slide. It's more than 15 years old now. And in this mathematical model, an immune cell and a non-immune cell that had a relationship were depicted as a line. And all of these different lines depict the complex interactions that occur. In fact, the authors point out at the time that the only networks that were then known to be as complex were the World Wide Web, and believe it or not, the social interactions of teenagers. Well, of course, those of you with teenagers will believe that very well. The key thing in this study was that we then have to think about how does a molecule like TNF sit as a pivotal regulator of such a complex network. Well, we've kind of moved away from the idea, therefore, that one cytokine could control all, and we think of teams of cytokines. And in this figure, this is an analysis that we put together 2 or 3 years ago, and in the top I'm depicting the cytokines that are essential for host defense against, for example, a gram-negative bacterial infection. So here we need TNF, IL-1, GM-CSF and IL-6 for effective host defense. If you look at the bottom left-hand side, what are the cytokines that you've got to have to mediate rheumatoid arthritis? Well, you need TNF, you need IL-6 and you need GM-CSF. You don't actually need IL-1, even though IL-1 is there. It's just not utterly critical. On the bottom right-hand side, we're now showing the same model for psoriasis, but it turns out you still need TNF, but you don't need IL-6 or IL-1. You do need, however, IL-17. And that's the way we start to think about functional pathways. As an analogy, here is the airline network of the world. So these are all the different routes that you can go around the world in a plane, or at least you could in the olden days. This is a snowman. Well, why am I telling you about snow in the middle of the Northern Hemisphere summer, at least towards the end of it? Partly because I'm in Scotland and we don't do summer very well, and secondly because when it snows in Glasgow Airport, the airline industry just simply don't care. Planes continue to fly around Europe. Glasgow's a great place, but it's not a pivotal node in the airline network. If there's even a centimetre of snow in Heathrow or Schiphol, the European air network stops. That's because Heathrow and Schiphol are pivotal functional nodes. If you translate that thinking across to immune therapeutics, we've now discovered that by blocking certain cytokines, TNF, IL-6 and IL-17 in different diseases, these are pivotal targets and as such, when inhibited, will lead to the entire immune response switching off and the patient's clinical disease improving. Now we can do this with more than just cytokines. We can do this, for example, with checkpoint modulators or with depleting cells, with B-cell depleting factors that we borrowed from the cancer literature. We can also now do this with small molecules that reproduce, if you like, the extracellular effects of these large biologic inhibitors and, in fact, are now entering immune disease therapeutics with impunity. So that's your cytokine and biologic primer for the day over. Let's go back to COVID. So remember, we started with an immune response on the left-hand side, which is an eradicating response. It's driven by type 1 interferons that lead to particularly cytotoxic T cells, killing virally infected cells, minimal lung damage, and the patient moves on. On the right-hand side in this pictorial alveolus, we're now depicting a situation where there's exaggerated immune responsiveness, that leading to high levels of cytokines, particularly TNF, IL-1 and IL-6, leading to tissue damage. The question is obviously now, can we use the same principle as we found in rheumatology to suppress responses in COVID? Well, the first clue that this might be possible comes from the recovery study, a very large trial still enrolling in the United Kingdom, in which dexamethasone was given as part of a controlled open label randomized trial. The outcome of this, I suspect, is well known to you. In an all-comer analysis, patients receiving dexamethasone were less likely to die, shown in panel A, and this was particularly evident in panel B in those undertaking invasive mechanical ventilation. If you look actually at those patients requiring oxygen only, there was still a benefit, and there was no benefit at all in those patients not receiving oxygen. In fact, if anything, the situation was slightly in reverse, and we don't really know what that means. But the conclusion here is that immune modification can potentially add value. Does it need to be just steroids? Well, the answer might be that we can use more specific interventions. We can go, instead of wiping out the entire airline network with steroids, we could go in thinking more specifically. And here's a study published very recently in The Lancet in which tocilizumab was analysed. It had been given to a number of patients in the ICU setting. This is a retrospective analysis, and you'll see that those patients receiving tocilizumab were more likely to survive. I would, however, caution you here because we don't yet know whether this is the real story. Randomized controlled trials in which tocilizumab is being tested against placebo have not yet been shown to be positive, and I think we just need to watch this space. It may be we need to choose patients more carefully. It may be we need to time the administration of drugs more carefully. Let us watch this space carefully. Tocilizumab is an IL-6 receptor blocker. It's not the only cytokine blocker we have available. There are clinical trials currently testing, for example, IL-1 inhibition, IL-18 inhibition, GM-CSF inhibition, a phase 3 trial ongoing based on an idea that GM-CSF sits as a fundamental regulator of innate immune cells. And remember, those were the cells that were high in patients who did badly. Here's a cartoon from an article we published recently in Lancet Respiratory Medicine showing the role that GM-CSF may have in activating macrophages, particularly in the lung. Now, I want to just quickly digress into the world of rheumatology again. The rheumatoid pathogenesis is driven mainly by activated macrophages, shown here in the top left-hand panel. These activated macrophages perpetuate damage to cartilage and bone and the release of cytokines, which have a, if you like, a virtuous cycle of destruction. And in health, the macrophage population in the joint is dramatically changed. And this is work that we recently published in Nature Medicine, led by Stefano Alvarenini in Roma, also part of my own group. And what we've done recently, and this work led by Mariela Krowska-Stolarska, we've compared the macrophages using single-cell sequencing in the rheumatoid joint with those in macrophages in the COVID lung. And this is what we found. We found remarkably that in bronchoalveolar lavage, there are populations of cells that are on the left-hand side, the blue rheumatoid macrophages and this green and purple alveolar macrophage were present, and we also found inflamed macrophages in the joints reproduced in the BAL of the patients with COVID, the red and the green cell here. But what we found remarkably was that when patients had higher numbers of these inflamed cells that we had recognized in the rheumatoid lung, their outcome was poorer. And this, of course, begs the question whether we could start to select the bad macrophages and specifically modify these, leaving tissue restorative, tissue repair macrophages present in the COVID lung. This is a really exciting possibility for the future. What about the comorbidities that have turned out to be so interesting and so tragically important in clinical disease outcome? We're now very familiar with the idea that patients who are older, but particularly patients who are obese, patients with hypertension and potentially with metabolic syndrome, are at considerably higher risk of severe disease and mortality. Why might that be? Could this give us a clue as to potential immune mechanism? Well, the truth is I don't know the answer to that yet, but I would wish to point out to you that obese adipose tissue is a highly inflamed place. In the cartoon on the left-hand side, you see here the obese adipose tissue full of activated macrophages. These same activated macrophages, by the way, that we have found in the rheumatoid synovial tissue and the COVID lung. A common theme coming together that innate immune activation seems to be a trigger for bad things happening. And of course, if you are in turn obese, then your rheumatoid arthritis is worse. And that might be because many of the factors that are present in adipose tissue in obese people are likely to be drivers of inflammation. Toll-like receptor agonists, these are sensors of damage in the immune response. Hypoxia, a driver of damage. Mechanical stress, a driver of damage. And of course, you're now thinking about a patient in the intensive care unit who's hypoxic, who's subject to mechanical ventilation, and who has large-scale tissue breakdown, potentially leading to vascular activation, potentially leading to the coagulopathy and microvascular embolic diseases that we are seeing. Of course, this is all hypothesis. And a final comment as to the last part of the immune jigsaw puzzle, can we vaccinate? Well, the answer is we hope so. There are a large number of vaccines in development. This is accessed a few weeks ago. In fact, it probably is out of date. There are even more candidates in development. You can see a whole range of different types of vaccine in the left-hand side of the table. And in this particular table, a summary of particularly advanced vaccine programs. I think we're particularly excited about the Oxford AstraZeneca vaccine, but I have no preference here. I think we only hope that some or several of these will induce not only immunity, but protective immunity that can in due course be demonstrably shown to reduce either the prevalence and incidence of disease or the progression of disease to severe forms. Well, thank you for staying with me this long. I'm aware that immunology is a long way away from imaging, but my take-home messages, I hope, are rather clear, even if some of the immunobabble in the middle became a little opaque. We're still at an early stage in studying the COVID immune response. I think it's worth pointing out that the vast majority of people infected with this coronavirus actually have a curative immune response, have a relatively mild disease, and we hope carry on to live normal, long and happy lives. But what I've described to you is as a disease of immune dysregulation. Now, I don't know if it's a true cytokine storm in the dogmatic definition that is formed of that following the CAR T-cell story, or whether what we're really seeing is simply chaotic immune dysregulation following an insult with a background of comorbidities, obesity and others that lead to generalized immune dysregulation. That in turn leading to damage to the heart, damage to kidneys, damage to the endothelium and all that goes with that. Regardless, this raises the enticing possibility of immune-targeted interventions. We've already demonstrated that glucocorticoids offer value, but what about highly specific interventions that could potentially deliver the same or even greater value, but with reduced potential for toxicity? And of course, vaccination remains our long-term hope. Let me finish with a quotation from Eric Hoffer, a philosopher whose work I admire very much. He states that simplicity marks the end of a process of refining, whilst complexity marks a primitive stage. In some respects, he's describing our current understanding of the immune response in COVID. It will eventually be clear, but only when we really understand the lessons of the experimental medicine and larger intervention studies currently ongoing. Of course, he wasn't describing the immune system. He was describing Michelangelo's definitive work. And those of you who have been in the Sistine Chapel, although you're surrounded by exquisite artistic beauty, your eye is always drawn to that singular moment in the center of the ceiling where man and God come together. Midst complexity, we will eventually find immunological interventions that lead, we hope, to curative therapeutics in COVID. Thank you very much indeed for your attention.